everyone. Good evening to all of you. Welcome to the third day of rapid revision rounds. Am I audible to all of you? Is the screen is visible? Can you please confirm? Hello, Jaswant. Hello, Gnanashri. Nandini. Yes. Okay. So, we have already completed 2 days session, right? Unit 1 and Unit 2. So, how was the sessions? Was it very much fruitful to you? You were able to realize where you are lagging and where you are confident of? Yes? Can you please confirm? Okay. So today we are going to discuss 2 units together. Okay, that is two most important, one of the, what I can say, important units from the syllabus. That is Unit 3 and Unit 4. So, fundamental process, which is basically what we have: replication, transcription, translation, all those things we are having under fundamental process. So, when it comes to CSIR exam, mostly you can expect experimental kind of questions. Sometimes you may also expect direct questions as well. So, without wasting much of the time, let's start with Unit 3. Are you all guys ready to give the answers? Yes, Ramachandra, mall bio and cell signaling both. Shall we start with the session? Shall we start asking the question? Okay. So here comes the first question to you all. The following figure represents the growth curve of wild type E. coli that is grown in a medium containing both glucose and lactose. So you can see this is the picture. Okay, that is E. coli grown in a medium having both glucose and lactose. In E. coli, the catabolite repression of lactose by glucose has been explained by different levels of CAMP in presence as well as absence of glucose. So I guess you all are aware of the relationship between CAMP and glucose. Am I correct? And what about catabolite repression? Yes? Are you aware of these things? Yes? Okay. So this model was challenged and experiments show that catabolite repression occurred because the activity of lactose permease was inhibited in the presence of glucose. Now, considering this second model, you have to find out the growth pattern of a mutant E. coli that is having loss of function mutation in LAC-I gene, and they are growing in a medium that contains both glucose and lactose. So these are the 4 options or the 4 graphs, and you have to decide which one is the correct one. You understood the question, right? Here they are following the second model. So based on this second model, you have to decide the answer. Yes? Keep on trying. Yes, Sanjay? Nida? Plot 2? Okay. Wow, I can see majority of you are saying the correct answer. Yes, some more answers. Some more answers. Yes. Now, very good. So I can see maximum of you are opting for option 2. Let's check out the correct answer now. The correct answer, as you all mentioned, is 2. So can you say me the explanation for this? LAC-I. Basically, it indicates what? So those of you who has given the incorrect answer, for them, Lac I indicates what? It indicates the inhibitor. But here what it is mentioned? Loss of function mutation occurred in lac I gene. So if there is a loss of function mutation occurs in lac I gene, what does it leads to? Yes, what does it leads to? Yeah, very good. It leads to constitutive expression. That is, whether we have an inducer or present or not present, beta galactosides will be expressed at higher levels, right? But we have lactose present in the cell. So this lactose can also be broken down. But we have also mentioned that glucose is also present in the medium. But in the question, it is clearly mentioned the activity of lactose permease was inhibited in the presence of glucose. Right? So that means what? When lactose gets exhausted, then the bacterial growth stops. Got it? Is it clear to everyone? Yes? Shall we move to the second question? Okay. So here is the second question. In an experiment, it was observed that a protein was upregulated in the cancer tissue compared to that of a control tissue. Now that showed correlation with disease progression. Following are few possibilities that can explain the above observation. So here you have to read the sentences properly. A mutation could be located in the 3' UTR region of corresponding mRNA at a microRNA binding site. Statement 2. Mutation changes the conformation of protein that may result in better stability. Statement C. A mutation in the corresponding mRNA promotes ribosomes read through the termination codon, resulting in increased synthesis of the protein. The last statement, a mutation in the corresponding mRNA increase the stability of RNA due to change in secondary structure. Now you have to find out the correct combination of statements. See, in such questions, wherever you feel dilemmatic or where you are not able to get the answer, you can try eliminating the options. Try eliminating the options wherever you find it difficult. So there are times when we eliminate the options, we are able to grasp the correct answer. Read the sentences properly. Then find the combination. Yes, Antara? Kantimati? Got only 2 answers so far. I guess everyone is trying out. Okay. Yes, Nandini, Nida, Jagdish. Just read the sentences properly. By reading the sentence itself, you can get the answer. Yes, Abid, Gnanashri. Further answers? Okay, there is some confusion between option 2 and option 4. Majority of you, as I can see, opting for 4, followed to that option 2, and some of you are also giving the answer 3. So let's find out what is the correct answer for the statement. But before going to that, I just want to ask you one question over here. Okay? Here one statement it is mentioned. A mutation in the corresponding mRNA promotes ribosome read through the termination codon. So if we have a protein and it reaches the termination codon, will there is any chance of increase in the synthesis of proteins? Can you say that? If we have a protein, suppose this is your polypeptide. Instead of terminating here, it will be terminating here. So do you think if it is upon termination here, there will be increase in the protein synthesis? No. So easily we can kick off this statement. So if this statement is removed, what we are having? A, B and D. So what is the correct answer over here? Fourth one. Right. So that is the statement C. What does it says? It will result in a different protein. Yes, there are chances it may result in some other form of protein, but it will never cause increase in the protein synthesis. Right? Okay. Now looking into the statement A, what does your statement A says? A mutation could be located in the 3' UTR region of corresponding mRNA at microRNA binding site. Now, why this option could be the correct one? You might be heard of a family of proteins or RNAs known as let7. It consists of around 12 microRNA members. Now, what is the purpose of these 12 microRNAs? They are responsible for acting as a tumor suppressor genes. Okay, so if they are acting as a tumor suppressor genes, what will happen? Suppose we have let7 and this will go and bind to the 3' UTR region of oncogenes like your RAS, MYC, anything. Then what happens? Definitely it leads to inhibition of their expression because they are tumor suppressors. Am I correct? But in that case, if this UTR site itself is mutated, then what happens? Definitely it leads to upregulation of oncogenes, right? So this leads to cancer. So that's why statement A is correct. Now coming to statement B and D, what does it shows? Statement B says that it will result in better stability. And statement D indicates what? There will be increase in the stability of RNA. So what you have to say about stability of a protein? How is it related? Can anyone say? Protein stability is determined by what? If you take a protein, what could be the factors that will determine its stability, its function? Yes? Yes, Vaishali? Amrita, Sridevi? Anyone? What are the factors that contributes to the stability of an RNA? Yes, it could be bonding, it could be folding. Very good, Nandini. Structural integrity, structure, everything. Yes. So not only these factors, there is another important factor that contributes to the stability will be the decay rate. See, once the mRNA is degraded, then they will undergo some kind of protein and they can be used as a transcription factor further. Right. And not only that, we have many diseases in which we have found that due to some alterations in mRNA stability, it has led to cancer, heart diseases, as well as immune disorders. Okay, so hence a mutation in the corresponding mRNA may make it more stable for increased rate of translation. Could you get me this one? Everyone? Why we have statement A, B and D is correct? Yes, confirmation will also come, Aditi. Why we have A, B and D statements correct? Got it? Everyone, kindly confirm. Okay. Thank you for confirming, students. So we can move on to the third question. Now, what is the third question? Yes. So before going into this question, I just wanted to say you something. Every time CSIR will always ask questions based on comparison between A DNA, B DNA, and Z DNA. And there are many times we do the same mistakes. So once we are clear with the basic things about all the three DNAs, it's not going to be a burden for us because you can easily expect 4 marks from here. Just go through the previous year question papers. You can expect in the form of match the following questions, and you can also expect in the form of statement based questions. So, one more thing to be added over here, when you are approaching these kind of questions, it is better to write down in a piece of paper what you know about it. Meaning, you have to trust your own knowledge. What happens is that when you will read the statements here, the chances are that you will get confused. So, what you can do here, it is clearly mentioned A form and Z form. You can scribble down the important characters of them, or you just see here it is mentioned right-handed helix and they are asking for configuration. Just find the difference between these two. Write down in a notebook, write down in a piece of paper, and then look into the option. Then you will understand what is correct. But if you directly look into the options, that there are chances it may make you confused. Got it? So let me see how many of you are able to give this answer correctly. Right-handed double helix and anti-configuration present in aDNA. Okay. Left-handed helix with alternating sequence of G and C, and syn and anti-configuration present in the zDNA. Right? 1 and 2 are quite similar, change in the configuration. Here it is anti, here it is syn configuration. Here it is anti-configuration, here it is a combination of syn and anti. Got it? Coming to third and fourth. The difference here is first two options are right-handed helix. The last two options are left-handed helix. So first you decide which one is actually coming under A form of DNA, whether it is right or left-handed. Yes? Yes, I can see very good answers from all of you. All of you are really doing very great. And this is a question came in the year 2020. 4 marks question. Yes. So if you are confident that it is a right-handed helix, then obviously you can skip these two options. And now there is a tug of war between option 1 and option 2. Just find out which one. So all of you has given the correct answer. So the correct answer is your option 1. So for this, you should not get confused. Yes, Debanjan, aDNA is a right-handed helix. So this is a table which I took it from Watson. You can follow this and try to remember all the things, because mandatorily you will get a question from here. If not in 4 marks, definitely you'll get it in the form of 1 mark. 2 marks. Okay. So important things like rise per base pair, helical rotation, what are the base pairs per turn of the helix, pitch per turn of the helix. And even I still remember in the year 2017 or 16, they have also asked with respect to major and minor group. So try to remember this as well. Glycosyl bond formation. So remember this table. This table is very important from examination point of view. Refer to Watson for that. Okay. So shall we move on to the next question? The next question here is about replication. Replication, if you look in the earlier times, they used to ask very much experimental questions from here. Nowadays, they are coming with some direct form of questions. This is a recent question. Following statements have been made about removal of supercoiling that is produced by DNA unwinding at the replication fork. So these are the options over here. Yes, Antara, exactly. Yeah, you can also remember that as well. Sugar and pucker, yes. So what are the options over here? Accumulation of supercoils is the result of DNA helicase activity during unwinding of DNA. Problem of DNA supercoiling is valid only for circular chromosomes of bacteria but not for linear chromosome. Supercoiling of DNA is removed by topoisomerases by breaking either one or both the strands of DNA. Both topoisomerase 1 and 2 are involved in positive supercoiling. So which one is the correct answer over here? Read the sentences properly. In fact, this is also a kind of question wherein you can eliminate the option and remember the answer, like get the answer. Very easily you can eliminate one particular statement. Yes, I can see a lot of correct answers coming in. Very good. Yes, yes. So what is, I can see many of you, maximum of you has given the answer as option 3. So just wanted to ask you one question. Why B is eliminated over here? Yes, why B is eliminated over here? Because what could be the reason? Why B is eliminated over here? Because circular chromosomes or linear chromosomes, both of them are undergoing supercoiling, right? Because topoisomerase is present both in prokaryotes as well as in eukaryotes. Do you remember the names of topoisomerase present in bacteria? Yes. Yes, Nandini. Yes, Ankita. Yes. Yes. What is the name of topoisomerase? Commonly said as gyrase. Yes. So that's why the option 3 is the correct answer. So DNA supercoils are generated during replication because DNA helicase eliminates the base pair between the strands. So upon elimination, what happens? Some kind of stress occurs. That stress is known as supercoiling. And as I said you before, it occurs both in circular as well as linear DNA. For resolving this issue, we are having the topoisomerases. So when it comes to type 1, it will cleave away one strand of DNA. And when it comes to type 2, it cleaves both the strands of DNA. Yes. And both of them are needed for resolving the positive supercoils. Okay. Fine. So we are moving to the last question of Unit 3. That is this one. This is the last year question. Given below are 4 sentences with labeled X, Y, and Z and L. RNA polymerase 1 transcribes X. MicroRNA are transcribed by Y. RNA polymerase is found only in plants Z. What is this TASIRNA? Can anyone say? What is this TASIRNAs? Any idea about it? Yes, TASIRNAs. These are synthesized by L. I'll come to that later. But before that, can anyone say what is TASIRNA is all about? Anyone? This is trans-acting small interfering RNA. This is trans-acting small interfering RNAs. Basically, they are needed for regulation. Okay, regulation of gene expression and all. And the machinery is usually carried out by the microRNAs, meaning you will have the same dicer complex, argonaut, RISC complex. Yes, exactly, Ria. Okay, so the processing and all is carried out by microRNAs only. Okay. Now this TASI-RNAs are synthesized by L. So what will be the correct answer for this? You have to find out X, Y, Z and L indicates what? Antara, TASI-RNAs means trans-acting siRNA, meaning they are responsible for some kind of regulations, regulating the stabilities, regulating the mobility. In fact, these are post-transcriptional modified RNAs. Okay. And what I can say, yeah, that much will be enough. These are mainly used for gene silencing, regulating the gene silencing mechanism, and everything, the processing, everything is carried out by microRNA based mechanism. That means you will have a dicer, you will have an argonaute complex, you will have RISC complex, finally leads to degradation. Got it, Antara? Okay, fine. So what will be the answer for this? Yes, I can see some mixed response over here. 4 and 3. So everyone has eliminated option 1 and option 2. So there is some kind of confusion going on between 3 and 4. Okay, yes. Yes, the correct answer will be answer 3. So what does 3 indicates? RNA polymerase 1, remember one thing, they are responsible for carrying out rRNA transcription. Okay. All these microRNAs, siRNAs, they are carried out by whom? RNA polymerase 2. Not able to answer, Ganashri? You will be able to. Just see. RNA polymerase that is found only in plants are of two types, that is 4 and 5. So definitely, first thing is that you can eliminate option 1 and 2. Right. Now what is left is option 3 and option 4. Now how you can eliminate option 4? Because here it is mentioned that Y, that is your microRNA, is transcribed by RNA polymerase 5. That is incorrect, because all the small RNAs like siRNAs, snRNAs, microRNAs, all these are transcribed by whom? Your RNA polymerase 2. Right. So as I already mentioned, siRNAs are transcribed by polymerase 2. That is also given here. So for this, you can remember this table. See, RNA polymerase 1, they are responsible for the transcription of majority of rRNAs, like the ribosome components, protein synthesis and all. Your mRNAs, snRNAs, siRNAs, microRNAs, all these RNAs are transcribed by whom? RNA polymerase 2. Coming to tRNAs, we have RNA polymerase 3. Could you get it now, Gyanashri? Others? Everyone has given, most of them has given the correct answer. Yes, Gyanashri, are you able to understand now? Just remember this table. Of course, plants is an exceptional case which contains only the polymerase 4 and polymerase 5. So you can remember like that. Remaining things you can remember from this table. This, I guess, this is taken from Lodish. So you can refer Lodish for this. So this is going to help you out. Okay? You are most welcome. So now we came to the end of Unit 3. Next. Yes, you all might be aware that we have started recently with Lakshya batch. So still some slots are available. You are most welcome. And we are like exams are coming near. So you might be studying hard. So this batch definitely going to help you in all aspects. It will provide you with 24/7 support system. It will provide you with the e-copy of Conceptica. It will also provide you access to your online study portal. Okay. So if you have something to be asked, you can call in this toll-free number, or you can visit our site stores.biotecnika.org. Okay. So make the wise decision, come to us. Okay. Next, another important. Yes, Sahiba. Yes, translation is DNA polymerase. Exactly. That is not RNA polymerase. DNA polymerase. Yeah. That is coming under your repair mechanism, Sahiba. Got it? Fine. So we are moving to another important unit, that is Unit 4, which includes the communication and signaling. See, this is basically a unit where you have to remember a lot of proteins name. Okay. Basically, if you go for a signaling, if you go for a cancer, if you go for cellular communication, if you go for immune system, anywhere you go, you will come across a lot of proteins. The only thing is that you have to find the connecting link between the proteins. Which protein comes under which category? That will help you in that. So shall we start with Unit 4? Are you all ready? And few things regarding Unit 3. Some concepts like translation, then regulation, all those things are also important. Even the comparative enzymes like prokaryotic enzymes are there, eukaryotic enzymes. Then you have got this inhibitors. All those things are important from examination point of view. Due to shortage of time, we cannot accumulate everything together. So we can straight away go to Unit 4 now. Now, the first question to you is, yes, during prolonged illumination, rhodopsin is desensitized, which leads to termination of visual response. The associated proteins are present in column A and their effects are given in column B. So you have to do the correct matching. But before going to that, can you say which pathway is this one? Yes, Debanjan, exactly. Can you say which signaling pathway is this one? What is the signaling pathway? The name of GPCR. Very good. But under GPCR, you have got a subdivision. What is that subdivision? Yeah, that I understood. GPCR, everyone is giving the answer. Under GPCR, which one is the one? Under GPCR, which is the pathway? Very good. Yes, Kriti, it is your GT pathway. So GT pathway is responsible for what? Is responsible for your vision. Right? Is responsible for vision. So now, find out the answer. No, Sahiba. Inhibitory is actually with respect to potassium channels. Okay? Yes, Antara. It is your rhodopsin protein. Yes, yes, yes. So, what is the answer for this question? Don't just blindly give the answer. Read this column, whatever the protein's name is present in the column A. Phosphorylated opsin, rhodopsin kinase, arrestin, phosphatase. What will be the answer for this question? Actually, this is again one of the favorite portion for CSIR. They usually ask questions from GPCR and more specifically speaking from GT. So this leads to a lot of confusion over here. You have to be clear with what happens during the dark time, what happens during the light conditions. So once you are clear with that, you'll be able to give the answer. Yes? Wow, everyone is giving the correct answer. Very good. I can see not a single incorrect answer. Yeah, one or two, but it's okay. You'll be learning something, right? Even if you're going wrong, no harm in that. You are learning something. Yes, very good. So, what happens here? What is the correct answer? The correct answer will be 3. So, what is here? Phosphorylated opsin. So, what is opsin? It is the protein portion of rhodopsin. So once opsin is phosphorylated, it leads to what? Decrease in the production of transducin. Why? Why it leads to decrease in the production of transducin? Can anyone say? If opsin is phosphorylated, why does it lead to decrease in the activation of transducin? Can anyone say? What could be the reason? I can see all of you are giving the correct answer. That's really good. Just one small query. Why this phosphorylated opsin leads to decreased activation of transducin? CGMP concentration decreases, that is another story, but that comes before to that. CGMP concentration will be coming after transducin activation, all those things, right? Yes, Nida? Because the rhodopsin, once it is activated, it leads to the activation of transducin. Because transducin is a G protein. So if rhodopsin itself is phosphorylated, how it is going to activate the transducin protein? Are you able to get me? So that's why that is the option. Rhodopsin kinase. Kinase means what? It is somewhere adding the phosphate. So definitely phosphorylating the opsin. Arrestin, that is needed for terminating the pathway. So it binds to phosphorylated opsin. Phosphatase means what? That removes the phosphate group. So this results in termination process. So that's why the option 3 is the correct answer. So this is a small flowchart for you which may help you in the exam. What happens during the dark and what happens during the light condition. Dark conditions, your CGMP level remains active. And this leads to what? Opening of cation specific channel. Once cation specific channels are open, it leads to action potential. So what happens? Our membrane becomes positively charged from inside. Right. And this leads to depolarization event, or we can say hypopolarization event. Ultimately, resulting in what? Release of inhibitory neurotransmitters. Can anyone give me an example of inhibitory neurotransmitter? Inhibitory neurotransmitter, can anyone give me an example for that? What is the example of inhibitory neurotransmitter? Inhibitory neurotransmitter, I am asking, Rimjim. GQ is a GPCR pathway. Yes, it will be your GABA glycine. So those neurotransmitters, if released, your bipolar and ganglionic cells will not be activated. These are the neuronal cells that are responsible for taking the information, transferring to the brain. So this leads to what? No vision. Yes, yes. Now, during the light conditions, what happens? We have CGMP getting converted to GMP. Can anyone say which enzyme is responsible for this conversion? Which enzyme is responsible for this conversion? CGMP to GMP conversion. Which enzyme catalyzes the reaction? Can anyone say? Which enzyme catalyzes the reaction of CGMP to GMP? Very good. Yes, Sahiba, this is your phosphodiesterase. PDE, phosphodiesterase. That is responsible for the breaking of your second messengers. So this leads to what? Closing of cation channels. So action potential will not be there. So there will be positively charged ions will move outside. This leads to hyperpolarization. If hyperpolarization occurs, the membrane becomes negatively charged. This leads to the release of excitatory neurotransmitters, due to which the bipolar and ganglionic cells will become active. Understand? Does anyone needs explanation for this picture? Or shall I proceed ahead? This is the picture for your GT pathway. How exactly in light conditions the things are happening, what happens during the dark condition, and what happens during the termination. Do you want me to explain this, or you are able to understand this one? Because I have seen many of you has given the correct answer, so only I'm asking. If you want, I will explain it. If not, I will proceed. What do you want me to do? Okay, sure. So we have got a rhodopsin. Kritika, this picture I took it from Leninger. Okay. So we have got rhodopsin protein. Okay, rhodopsin, when the light hits the retina, rhodopsin protein gets activated. So within the rhodopsin protein, what happens? 11 cis retinal gets converted to all trans retinal. Okay. After that, once rhodopsin is active, this leads to the exchange of GDP to GTP on a transducin protein. Okay. So now your transducin is activated. After the activation of transducin, this will help in the removal of inhibitory subunit from phosphodiesterase. Okay. So this leads to what? This leads to activation of phosphodiesterase. Once the inhibitory subunit is removed, your phosphodiesterase will become active. Once they are active, what happens? Your second messengers will be degraded. Once the second messengers are degraded, your cation specific channels will close, and whatever the ions that are present inside will go outside. This leads to hyperpolarization event. Okay, this is what happens during the light condition. So when we are entering into the dark condition, what we are having? Already we are having low calcium level. Now, this low calcium level leads to what, it leads to two conditions. First, it inhibits phosphodiesterase. Second, it activates GC. Can anyone say me what could be the reason behind this? When we enter into the dark condition, the the cytosolic calcium levels are low. The low calcium levels will inhibit your phosphodiesterase and activates your GC. What could be the reason behind it? Can anyone say? Because GC will lead to what? Lead to the activation of CGMP. CGMP will lead to what? Opening of channels. Am I correct? So in order to prevent this, you have got phosphodiesterase. Because if phosphodiesterase is active, it will lead to the degradation of CGMP. So if CGMP will be degraded, channels will not be open. So that is not the case when we enter into the dark. That's the reason your low calcium level inhibits your phosphodiesterase and activates your GC, thereby opening the channels. Now, light condition is over, dark condition is over. Now, how you are terminating the pathway? For termination, you have got rhodopsin kinase. This rhodopsin kinase will go and phosphorylate your rhodopsin protein. And once they are phosphorylated, they are attracted by another protein known as arrestin. So arrestin will also go and bind over here. So after that, what happens? Slowly after some time, the arrestin will be dissociated and whatever the phosphate is present within the rhodopsin molecule will be removed, or you can say dephosphorylated. So during that time, all the trans retinal will be removed and all the cis retinal will come inside. So what happens? Your rhodopsin molecule is ready to take up another step of signal transduction pathway. This is what your GT pathway is. Is it clear to everyone? Whoever was asking for explanation, Swarnadeep, Kritika, Sridevi, is it clear? Or having any doubt? This is very important pathway from examination point of view. GPCR itself is very important, but among GPCR, this is very important because here you can expect some kind of twisted questions of dark and light conditions. Okay. So please remember this one. This is going to help you. So quickly moving on to the second question, that is with respect to apoptosis. So, apoptosis in C. elegans, activation of SET3 and SET4 are essential for the apoptotic pathway. In addition, gain of function mutations in SET9 gene causes this protein to be made in cells that would normally die but results in the survival of those cells. Right? Given these facts, which one of the following diagram correctly represents a cell death pathway? So you have 4 different images, and among these images, which one is the correct answer for a cell death pathway? Can you say? Yes. What is the correct answer for this question? You may think for some time. See, in such cases also, like I said you before, in a rough paper, you can draw the flow chart which you know, right? Which you have studied, which you believe in yourself, which you believe in your studies. So which one will be the correct answer? Find it out. This was a recent question. And you know what? This C. elegans based questions are kept on asking every time in the exam. Yes, Vishnu, Nandini, Riya, Raumi, Aditi, Ritika, Vishal, Ankita, Amala. Yes. So the correct answer will be what? The correct answer will be the 4th one. So what is this EGL1? Anyone can say? What is this EGL1? What is this EGL1? So definitely you can eliminate this option because it leads to survival. But here, what is the question asked? Cell death pathway. So from option 1, 2 and 4, you can select one, but you have already selected the correct answer. What is this EGL1? What is the EGL1? Who is inhibiting SET9? Yes, I'm getting the correct answer. Very good. Can anyone say what is this EGL1? And how it is inhibiting SET9? What is the role of EGL1? Okay, I will show you the picture. It will be more clear to you. We have got 3 different types of proteins here. Regulatory protein, adapter protein and effector protein. Right? So, SET9 becomes the regulatory gene. And SET4 becomes the adapter. So when you don't have a stimulation, SET9 will be inhibiting the SET4. So there will be no SET3 activity and it leads to cell survival. Yes, as you said, when EGL1, a kind of pro-apoptotic stimuli will come, what happens? SET9 will be inhibited. And this leads to the activation of SET3, finally leading to cell death. Similar to that, in vertebrates, we are having SET9 analogous to BCL2, SET4 analogous to APAF1, SET3 analogous to caspase-3. Okay, remember the analogous proteins present in vertebrates. They may ask in the question. And this is yet another group of, another table which provides you with the examples of pro-survival members, that is responsible for cell survival, which includes your BCL2, XL and all. Then those that includes pro-apoptotic members that induces apoptosis. That is broadly classified into 2 types. One is channel forming proteins, another is BH3 only proteins. It is this BH3 only proteins that act as a signal for a cell to undergo apoptosis. Okay. Yes. Now quickly moving on to the 8th question. So we have got a Western blot analysis after treating the cancer cells with a prospective anti-cancer drug. So these are the assumptions that is being made. The drug may have arrested the growth of cells at the G1 phase. The drug might have targeted the JAK-STAT signaling pathway. The drug led to the apoptosis of the cell. And the last option is drug-induced apoptosis was through the extrinsic, or you can say mitochondrial independent pathway. So look into the picture, find out the correct combination. Read the options, analyze this Western blot, then get the answer. Yes. Check what are the proteins that are being involved. Sahiba Nida has given the answer. What about others? Read the assumptions clearly. The drug hath arrested the growth of cells at G1 phase. The drug might have targeted the JAK-STAT pathway. The drug has led to the apoptosis of the cell. So from this protein's name, which you can see, which indicates that the cell has undergone apoptosis? What indicates that the cell has underwent apoptosis? Yes, Ritika, Nandini, Subhash. Yes, very good, Ritika. Got your answer. What about others? Among these proteins, which is the indicator for apoptosis, it will be a cleaved part. Always remember one thing: two markers are there for a cell that has undergone apoptosis. One is cleaved part, and what will be the second one? What will be the second one? Can anyone say? What is the second indicator of a cell that has underwent apoptosis? Second indicator that signifies the cell has underwent apoptosis. Can anyone say? Yeah, answer, I got it. Many of you has given 2. Many of you has said as 3, 4. Not cytochrome C. Because cytochrome C, if you say, it will be under the category of intrinsic pathway. Yes, very good. Phosphatidylserine, which is normally located in the inner membrane, upon apoptosis, it will be translocated to the outer membrane. That is another indicator of your cell that has underwent an apoptosis. Okay? Yeah, exactly. Aditi, Sahiba. Correct. So, the correct answer will be 2. Now, why? I have already mentioned. So, cleaved part is the marker for apoptosis, right? And you are very well aware that G1 phase proteins like CDK6 and cyclines are there, which is accelerating your G1 phase. And they are the one which is responsible for phosphorylation and regulation of RB gene. You know the connecting link between RB gene and the CDK cycline, right? Because if you have cyclin CDK protein, cyclin CDK is the one that is responsible for phosphorylating your RB gene. So once your RB gene is phosphorylated, this leads to what? Cell cycle progression. If you remember, one pathway is there, right? So since we have seen there is concentration of CDK6 and cyclin D1 is accumulating, the cell cycle may be arrested. So that's what shown in the graph. If you look into the graph, CDK6 and cyclin D1, you are having much thick bands. So that is the accumulation. But when you see the JAK-STAT proteins, that is STAT3 and p-STAT, these are the products of JAK-STAT pathway. Here the bands are thin. Why? Because the drug might have targeted this pathway. That's why you're not getting the bands over here properly. Right. And the last option which is being given, drug-induced apoptosis was through the extrinsic or mitochondrial independent pathway. We cannot say that. It has underwent apoptosis, that is fine. But how exactly it went apoptosis, we cannot say that. Okay. So that's why D is quite unsure. That's why option A, B, C is the correct answer. Okay. So this is the 3 different pathways of apoptosis. Of course, caspase 3 becomes the most common pathway, common caspase or the executional caspase for all the three pathways. But when it comes to initiator caspase, for extrinsics, it is 8. For intrinsic, it is 9. And for perforin, it is caspase 10. Remember this one. This flowchart is important from examination point of view. Try to remember this one. Okay, so moving on to the next question. Cellular levels of tumor suppressor protein P53 is maintained by ubiquitin ligase protein MDM2. Overexpression of MDM2 was found to convert a normal cell into a cancer cell, thereby destabilizing P53. Another protein inhibits the activity of MDM2, thus stabilizing P53. Loss of P19ARF converts normal cells into cancer cells. Now you have to find out the correct combination of treatment. Nida, CDK6, it is targeting your G1 phase proteins, right? That's why it leads to the accumulation. So if G1 phase is being halted, how the cell cycle will be proceeding? Are you able to get me, Nida? Nida Praveen? In the picture, it was given that drug is targeting your G1 phase proteins. That's why it got accumulated over there. Got it? Are you able to get me, Nida? Yes, what could be the answer for this one? Yes. So before going to that, you have to understand what is this relationship of MDM2 with P53. I hope so many of you might be aware. MDM2 is a ubiquitin ligase that helps in inhibiting the P53 and keeps the P53 low in the cytoplasm. So only when it is needed, it will be activated, right? So here they have said overexpression. Overexpression is something related to gain of function mutation that led to cancer cell. Loss of function mutation led to cancer cell. So that is an indication of what? We have tumor suppressor genes present because it is your oncogenes that are responsible for undergoing gain of function mutation, and it is your tumor suppressor genes that is responsible for undergoing what? Loss of function mutations. Among this, which one is the dominant one? Oncogenes mutations or proto-oncogene mutation or tumor suppressor gene mutation? Which one is dominant? Yes, the answer is correct. Which one is dominant? Oncogene. The proto-oncogene is mutation is dominant, or the tumor suppressor gene mutation is dominant? Proto-oncogene. Yes. Why we have proto-oncogene as the dominant one? Because a single allele mutation will be enough to convert a proto-oncogene to oncogene. But when it comes to tumor suppressor gene, what happens? We have to know that it is recessive. Why? Because if both the alleles are being lost, then only it leads to cancer. Yes, exactly, Sahiba. Very good. So moving on to the last question. A researcher was studying a protein X which was being absorbed to move across cells when an extracellular electrical stimulus is provided. An artificial peptide P was prepared which resembles the structure of connexin and competitively inhibits the connexin formation. So you have to find out the fate of protein X if the cells are treated with peptide P and then if the electrical stimulus is provided. X fails to move across the cells due to improper formation of tight junction, or due to the formation of gap junction, or due to the improper formation of desmosomes, or X moves freely across the cell. What could be the option? What could be the correct answer for this question? Yes, Ritika has already given the answer. Nida? Yes? Yes. All you need to do is you have to remember the protein name. If this protein comes under which category, if you remember, you will be able to answer this kind of question. Cellular communication is a very easy chapter, only if you remember the protein's name. If I say occludin, what junction it is? If I say occludin, what is the junction here? This is connection. Why do you call connection? Because gap junctions are formed by 6 different connection proteins. Right? Yes. It comes under the category of tight junctions. So you have to remember the junctions and the corresponding proteins. So that is your answer to connections that make up the gap junction. So P being a disruptor of connection formation, it will not allow proper gap junction to form, and hence the protein X will not be able to travel between the cells. So I am providing you with another table. This is again very important table from examination point of view. You can easily segregate cell to cell interaction and cell to matrix interaction. Corresponding to that, you will have the receptors, the cytoskeletal proteins, the adapter proteins, and the corresponding function. The problem here is we will get confused with the adapter proteins that are being involved and the cadherin sometimes. That is the receptors. So be clear with this table as a whole. If you are clear with this table, you are definitely going to get good marks for cellular communication. Of course, apart from that, you have got cell adhesion molecules structure. And this cell adhesion molecule structure is very important. Again, that is not given in the table, but just reminding you, fibronectin, laminin, integrin, their signaling, all those things will be very important from examination point of view apart from this. So students, how was the session for today? You are confident now that you are able to solve the questions? Yes? All the questions from, of course, there are many topics which is yet to be touched. You have to do a continuous revision on a regular basis. You have to spend time on this revision as well as on practicing the questions. Okay, so please do practice MCQs and uh, revise the portions on a regular basis. Thank you so much. Wishing you all the best for the upcoming exam. You all are definitely having good energy and you can achieve this your goal. Okay, thank you so much.